All right, here's the second part for Lecture 5, Part 3. Wrapping up our discussions of metabolism, we're going to focus now on glycolysis and fermentation. The goals for this are that once you go through this lecture and you take notes and you do the accompanying reading in your textbook, which is Chapter 4, you should be able to define fermentation in biochemical terms and understand how the term can in fact have multiple meanings, even all within the context of microbiology. You should understand the stages of glycolysis and what is happening at a basic level in each of those stages. You should understand the importance of stage 3 in the context of microbiology and know when it happens, like under what cellular conditions, and what the purposes are of stage 3 from a biochemical perspective. And you should be familiar with the different examples of fermentation products that exist and their applications for human beings. Glycolysis, which is also known as the Embden-Meyerhof pathway in honor of the discoverers, serves many microbial cells as the first stage of glucose metabolism in the ultimate process of respiration, whether it be aerobic or anaerobic respiration. And so I think that's the context in which most of you are familiar with glycolysis. It's in studying the overall process of aerobic respiration in your earlier coursework. It's important to remember that glycolysis is actually one of many possible types of fermentation. And a fermentation means, biochemically, that the same substance, which is typically an organic molecule, is both the electron donor and acceptor. These are catabolic processes. We have an internal electron donor, and the same substance, albeit somewhat converted into a metabolite of the original substance, is the electron donor, and then later the electron acceptor. So we say that that then is an endogenous electron acceptor. Some fermentative organisms are capable of both fermentation and respiration, and so they will use their environmental conditions to dictate when they ferment entirely as a means of making ATP, or when they use that as a precursor and actually do respiration. And by precursor, I mean the first of 3 uber stages of respiration. So often the case will be that a microbe that can both ferment and respire will choose to respire if there is available terminal electron acceptor. For example, if there's oxygen in the environment, if it's an aerobic respirer. However, there are other environmental cues that could trigger a microbe to go between fermentation or respiration. For example, the yeasts that are used in ethanolic or alcohol-yielding fermentations are triggered to ferment instead of respiring, both by reduced oxygen in the environment, but also by some of the chemicals that are found in hops. Hops are a particular plant flower that is used in beer production for flavoring and also to trigger the yeast so that they ferment sugars into alcohol rather than taking their sugars and driving them through aerobic respiration and yielding no alcohol. So in total there are 3 stages of glycolysis, and this is the fermentation process we're going to think most closely about in this lecture. There are 3 stages, but in total when they're done, what happens is you have 1 glucose go in and you have 2 ATP coming out. The ATP in fermentation are always made using substrate level phosphorylation. Okay? So when not associated with respiration, glycolysis will result also in the production of fermentation products. For example, many yeast will produce ethanol, and the lactic acid bacteria will produce lactate as a result. These fermentation products have to be made so that NAD+, the oxidized form of NAD, can be regenerated, allowing the organism to continue running substrate-level phosphorylation. Now we're going to walk through and break apart everything that I just said. So there are 3 stages to glycolysis, and here we're looking at what happens in stages 1 and 2. In a respiratory organism, if respiration is occurring, then that's actually all that happens is stage 1 and 2, and then the results, which is pyruvate, will go off to the tricarboxylic acid, or TCA, or Krebs cycle, and then ultimately will carry on to the electron transport chain. But first let's think about stages 1 and 2. So stage 1, which is also called the preparatory reaction, begins with glucose and ends with glyceraldehyde 3-phosphate. So it would end over here. That would be stage 1. And then stage 2 would be everything that's happening down here. So this would be 2, this would be 1. Okay? So the stage 1 is also often called the preparatory reactions. And in that, glucose is phosphorylated by ATP. That's catalyzed by hexokinase over here. So we're putting an ATP in, and the result is glucose 6-phosphate. This is an activated or higher energy molecule. That is then rearranged by the enzyme phosphoglucose isomerase. This is a rearrangement, okay, into the molecule fructose 6-phosphate. An additional ATP goes in, a phosphofructokinase catalyzes that reaction, so now we have 1 and 2 phosphate groups, and we have the molecule fructose 1,6-bisphosphate. That is then reacted upon with the help of the enzyme fructose bisphosphate aldolase, and it yields two isomers, 2 3-carbon isomers, each with a phosphate group. The first is dihydroxyacetone phosphate, and the second is glyceraldehyde 3-phosphate, often abbreviated G3P. Okay? Now, these are isomers of one another. There's an enzyme that can convert one to the other, but in glycolysis it's the G3P, or glyceraldehyde 3-phosphate, that actually continues on into stage 2 of glycolysis. And that is actually where redox reactions begin. All of these other reactions in stage 1 are not redox reactions. There are different types of reactions, rearrangements and phosphorylations. So in stage 2, now we see enter electron carrier. So there's the oxidized form of nicotinamide adenine dinucleotide, and the first thing that will happen in stage 3 is that NAD+, the oxidized form, will be reduced to form NADH. A proton will be yielded from that reaction as well. This is going to happen 2 times for every 1 glucose molecule. Glucose has 6 carbons, glyceraldehyde 3-phosphate has 3 carbons. So in order to account for each 1 glucose in the overall glycolytic pathway, you have to have that reaction, you have to have all the stage 2s happening twice. Okay? So we oxidize NAD+ and we get NADH. That's a redox reaction. Okay? The result of it is 1,3-bisphosphoglycerate as well as an NADH. Okay? The 1,3-bisphosphoglycerate is then used as a provider of a phosphate group. So this phosphate group is going to actually be added to ADP to form ATP, an enzyme, the enzyme that catalyzes that is phosphoglycerate kinase. So here we have substrate level phosphorylation. Okay? When ADP receives a phosphate group from an internal organic molecule, and an enzyme simply transfers that phosphate group onto ADP, making ATP. There's no ATP synthase, there's no proton motive force, this is just a chemical reaction catalyzed by one enzyme. Okay? The result then is 3-phosphoglycerate plus that ATP. The 3-phosphoglycerate is then rearranged by a mutase enzyme into 2-phosphoglycerate. That's then acted upon by an enolase. Water is released, okay, and we form a high energy molecule called phosphoenol pyruvate, or PEP. You've seen that molecule in earlier classes. The PEP is then again used as the provider of a phosphate group. So that phosphate group is used to conduct substrate level phosphorylation, okay, that's catalyzed by pyruvate kinase. An ATP is released, and we're left with the 3-carbon molecule pyruvate. Remember, all of this is happening 2 times, so here we would actually have 2 ATP, and here we would have 2 ATP. Up here we put 2 ATP in, so 2 ATP went in, 4 ATP came out in stage 2, so we have a net gain of 2 ATP due to substrate level phosphorylation. Okay, we've also made NADH, and in respiratory organisms, if they're doing respiration, that could go off to the electron transport chain. Okay, if it's a fermentative organism, it's going to go on to stage 3. Okay, so let's consider stage 3, because this part of this lecture is about fermentation, not about respiration. So again, once you complete stages 1 and 2 of glycolysis in respiratory organisms, you would then move on and talk about the Krebs cycle. All of that pyruvate would go off to the Krebs cycle. So here's the Krebs cycle over here. So this pathway, or this image, is based on an aerobic respiratory organism that can also ferment. Classic examples of that would be yeast, like S. cerevisiae, okay, or lactic acid bacteria, or humans. Okay, so if there's oxygen present in organisms like that, aerobic respiration will ensue. Those pyruvate will be converted to acetyl-CoA. It will enter the Krebs cycle, and the Krebs cycle will fully oxidize those pyruvates, creating more NADH and FADH2 as well. They will go off to the electron transport chain and contribute to the production of a proton motive force, which ATP synthase will harness to make ATP using oxidative phosphorylation. All of the carbons will be fully oxidized, and they'll be found in the highly oxidized molecule CO2, which comes out of the Krebs cycle. But this is about fermentation. So let's think about what might happen in our own cells, or in the cells of lactic acid bacteria, or in the cells of many yeasts, such as Saccharomyces cerevisiae. So in those organisms, when there's no oxygen present, they will go on to stage 3 of glycolysis. And stage 3 of glycolysis is where alcoholic fermentation or lactic acid fermentation occurs. Alcoholic fermentation results in the production of alcohol, ethyl alcohol, and CO2. Lactic acid fermentation in the production of lactic acids. The point here is that you get acids and gases. And here what we're seeing is stage 3 of glycolysis. Okay, let's consider that in more detail. Shown here is an ethanol or alcoholic fermentation resulting in ethanol and carbon dioxide as waste products. CO2, which would be bubbles. So that would be carbonation in your alcoholic beverage, or it would be the bubbles that allow bread to rise if you're making bread. Many of the yeasts in the Saccharomyces cerevisiae group can do this particular fermentation shown. The important thing to notice here is that NAD+, okay, is regenerated, and that is done by oxidizing NADH. So pyruvate came out of stage 2, right? That's where we left off in those earlier discussions of stages 1 and 2. The pyruvate is acting as an electron acceptor. Remember that biochemically speaking, fermentation means that you basically have the same molecule acting as the electron donor and the electron acceptor. Glucose is the donor in these organisms when they're fermenting, and pyruvate is the acceptor. Because all those carbons in pyruvate actually came from glucose, functionally speaking, these are the same molecules here. Okay, so the pyruvate is then going to be used as the electron acceptor. It's going to accept the electrons inherent to this hydrogen here in NADH, regenerating NAD plus and creating ethanol and CO2 during the metabolic steps that are part of alcohol or ethanolic fermentation. That NAD plus is then able to return, okay, to stage 2 of glycolysis. And so what this is showing up here, this is showing stages 1 and 2. It's pretty messy, sorry. Stages 1 and 2 of glycolysis, just all summed up in one arrow, and we're getting a net gain of 2 ATP. Okay, so that won't happen unless you have NAD plus, in which is going to act as the electron acceptor, removing electrons from glyceraldehyde 3-phosphate, as I showed you in the earlier figure. So you have to regenerate this NAD plus in order to continue running stages 1 and 2 and making ATP. And if the organism is relying on substrate level phosphorylation, which is occurring during glycolysis, to derive ATP so that it can do all of its metabolisms and its anabolisms and its cell division, then you've got to keep regenerating the NAD plus to run that process. And you do that by dumping electrons to pyruvate, which will convert it into ethanol and release some CO2 along the way. Now, in human cells or other vertebrates in the muscle tissue, when we are undergoing oxygen deprivation, for example, during prolonged exercise, or in lactic acid bacteria, which we use to make yogurt, okay, they do a slightly different type of fermentation, so they have different enzymes that are acting during stage 3 of glycolysis. So again, up here, we've got stages 1 and 2, net gain of 2 ATP in its annoying energy ball form, and out of that comes pyruvate. Okay, the pyruvate is again serving as the electron acceptor. Okay, and the electron, and when it accepts those electrons, it is taking them from NADH, which will regenerate NAD+, which can then go off and continue participating in stages 1 and 2 of glycolysis, specifically stage 2, allowing those stages to continue running and allowing continued ATP production. Okay, the pyruvate, when it acts as an electron acceptor in these organisms and in ourselves, is converted to lactic acid or lactate, shown here. Okay, each 2 pyruvates to 2 lactic acids, that's 1 each. All right, and that's very acidic, so it equals sore muscles in us, or spoiled milk when lactic acid bacteria are doing this in milk, and that spoiled milk curdles and forms yogurt. So the whole point of this from the point of view of the organism is to generate more ATP by coming up with a means of regenerating oxidized nicotinamide adenine dinucleotide. So we could have an entire course that focused on fermentations, on the microbes that do the fermentations, on their biochemistry, genetics, and ecology, and how those fermentations have been used by humans today and in the past, and how that's affected human culture. But we don't really have time for that. I do, however, want to mention the Clostridium and some of the fermentations that they're capable of doing. I've mentioned Clostridium organisms, Clostridium species earlier in class. I've mentioned the pathogens. Clostridium difficile causes enterogastroenteritis, also Clostridium botulinum and Clostridium tetani, which cause botulism and tetanus respectively. But there are more desirable Clostridium out there, and some of them are particularly useful for humans in that they can do fermentations that result in end products that are useful to people. Ethanol, acetone, butanol, and butyrate, as well as acetate in particular. Acetone and butanol in particular are heavily used in industrial applications. And today we get most of those materials from refining petroleum. But during World War II, when there were petroleum shortages, humans relied on Clostridium and their ability to make them when they fermented glucose. And I expect with the green chemistry movements afoot today that and also the potential petroleum shortages and how difficult it is to get petroleum out of the earth these days, that we're going to see a resurgence in the application of Clostridium to derive these useful end products. The term fermentation can be used in a variety of ways, and this can be quite confusing. In this lecture, we've focused on using the term according to its biochemical definition, strictly referring to the use of an endogenous, or internal electron donor and acceptor, which are ultimately the same molecule, which is being metabolized progressively. For example, glucose as the donor, pyruvate as the acceptor. However, in the food and beverage industry, the term fermentation can mean more than this. It can be a term used to refer to the use of microbes to produce molecules that give food characteristic flavor and sometimes texture. The number of foods and beverages we routinely consume that are fermented, if you use that definition, or in other words have their flavoring and physical properties altered by microbial growth, is somewhat staggering. This list includes chocolate, coffee, tea, salami, hundreds of fermented vegetables, pickled eggs, and so on and so on. It's a lot more than just wine, beer, cheese, and bread. In fact, it's likely that every one of you consumed at least one food or beverage today that has benefited from microbial fermentation. From a historical context, you should also be aware that fermenting food is something that can increase its nutritional value. For example, vitamins in cabbage are more available to us when the cabbage has been fermented into sauerkraut than they are when we eat raw cabbage or even after cooking cabbage. Likewise, fermented foods, because they already have a population of microbes growing in them, are less likely to support the growth of additional microbes. Thus, they are less susceptible to spoilage. This is why we humans have so many fermented foods, because in the days prior to refrigeration, we needed ways to make foods last, and we needed ways to make our food more nutritious than as we do now. So humans have evolved in all different parts of the world, culturally evolved, to ferment a variety of foods and beverages, and those play a very unique role from a cultural and sociological point for people. So with that, I'm going to conclude this part of this lecture, and you can then move on to Part C.